Okay, so welcome everyone to another journal club. Today we have the pleasure of having Loïck Ducros, who is finishing his PhD in Simon Lamar and Nico Pichot's lab in University of Moncton, Canada. And he will be talking about his paper just accepted. I didn't find the final proofs corrected version, but it just came out last month in JEB about Arctic char and cyclic hypoxia. So the floor is yours.
So thank you, Enrique, and the committee for the invitation. So everybody see the slide? Do you see the slide?
Yeah. It's okay.
Okay.
We can see
it. Perfect. So yeah, in fact, this paper is the second part of my PhD project. So my project focused on how Arctic char, a salmonid, respond to cyclic hypoxia. And with this paper, we try to understand what happened at the level of the metabolic rate and mitochondrial physiology. And so why cyclic hypoxia? As we know, the planet is not going well. And so we have an increase of cyclic hypoxia event. And these events are very stressful for the fish. In fact, during the hypoxic phase, due to the low oxygen availability, there is decrease of the ATP supply. And if the fish want to survive, they have to match the ATP demand with this decreased ATP supply. So there is a big stress about the maintain of the energy homeostasis. And during the reoxygenation, in fact, the fish have to limit its oxidative stress because during the hypoxic phase, we have an accumulation of the succinate inside the mitochondrial matrix. And during the reoxygenation, all this succinate is reduced into fumarate, and this could cause a reversal of the electron transport inside the mitochondria, which could produce an overproduction of ROS at the level of the complex 1, and so cause an oxidative burst. So as you can see, the fish have to adapt its aerobic metabolism to survive. So why I focus on Arctic char is because Arctic char is sensitive to low oxygen levels, and we don't know a lot about the effect of cyclic hypoxia on fish, and we don't know a lot about how hypoxia-sensitive fish are impacted by cyclic hypoxia. So during my PhD, I work on Arctic char, and I expose them up to 1 month of cyclic hypoxia. When I talk about cyclic hypoxia, that means that for 1 cycle, so 1 day, we have a normoxic phase during the day, and then at night, we have the hypoxic phase, followed by the reoxygenation. During the first part of my PhD, I found that the first days of cyclic hypoxia are very stressful for the fish. In fact, we saw a huge impact of their hepatic metabolome. There is a huge metabolome alteration. So we think that it's normal because it's an hypoxia-sensitive fish. However, after 10 days of cyclic hypoxia, we have a decrease of this metabolome alteration. And after 1 month, we have a complete homeostasis came back at the level of the hepatic metabolome. So that means that this fish, Arctic char, is sensitive to low oxygen level, but is able to acclimate to cyclic hypoxia. And we want to understand how these fish acclimate to cyclic hypoxia. And for this paper, our global hypothesis was that the acclimation to cyclic hypoxia requires a downregulation of the aerobic metabolic rate, particularly during the hypoxic phase. And then the fish have to upregulate its mitochondrial respiratory capacities to mitigate the constraints on aerobic metabolism and the elevated risk of oxidative stress. So this is the global design of this project. So we expose Arctic char up to 10 days of cyclic hypoxia, and we perform 3 experiments. So we measure the metabolic rate, the mitochondrial respiration, and the ROS level at the level of mitochondria. So for the first part of this project, so these experiments were performed by Anisara, and we measure, in fact, the MO2, the oxygen uptake, which is a proxy for the metabolic rate, in fish exposed to normoxia or cyclic hypoxia during 10 days. And so the first thing that what we observe is that For the standard metabolic rate, we have no differences between the normoxic and cyclic hypoxic group. So in fact, the fish defend their standard metabolic rate, and we observe no metabolic depression during the hypoxic phase. However, we also measure the sporadic increase of MO2. So as you can see here, you have the basal MO2, so what we use to measure the standard metabolic rate, but sometimes you have this sporadic increase of MO2. And if we focus during the night, at the level of the night, so that means it's during hypoxia for the cyclic hypoxic fish, what we found that in the normoxic group still have this sporadic increase of MO2. However, in the cyclic hypoxic group, during the hypoxic phase, we have this suppression of this sporadic increase of MO2. And this sporadic increase of MO2 are a proxy for spontaneous swimming activities. So when the fish have a little locomotion activity, we have this sporadic increase of MO2. So with this result, we show that during the hypoxic phase, fish didn't perform a metabolic depression. However, they suppress their activities to survive and so to save some energy, but they defend their standard metabolic activity. So that was for the first part of this project, and then we measure the mitochondrial respiration in 3 tissues. So we measure it in the permeabilized liver, heart, and brain. So we use for this, of course, an Ouroboros, and we characterize the different respiration state for these tissues. And we also measure the mitochondrial P50. The P50 is the oxygen partial pressure at which the respiratory fluid is at 50% of its maximum. So to measure it, in fact, we let the Ouroboros chamber to go into anoxia, and using a Michaelis-Menten model, we were able to determine this P50. And while we measure this P50, in fact, this P50 allows us to understand how the mitochondria perform under low oxygen level. So that means if you have a low mitochondrial P50, that means that the mitochondria is more efficient to work under low oxygen levels. So here, what we found for the 3 tissues. So first, for the liver, for this graph, so you have in blue the normoxic group, and in orange the cyclic hypoxic group. We measure the leak, OXPHOS, and COX at day 1, 3, and 10. And first, for the leak state, we observe a general trend of increase of the leak state in the cyclic hypoxia group. And this could be a strategy to prevent from ROS production. For the OXPHOS, we observe an increase of the OXPHOS during the first day of cyclic hypoxia. However, after 10 days, we have the homeostasis came back, and we no longer observe any difference between the cyclic hypoxic fish and the normoxic fish. However, due to the fact that we still have a high leak, we have a slight lower coupling in cyclic hypoxic fish after 10 days of hypoxia. I have a question here.
Yeah, just quick question. When the fish were sacrificed, is it during the nighttime phase or the daytime phase?
Oh, sorry for this. We sample the fish after the reoxygenation.
Okay, thank you.
Okay. So that was the result for mitochondrial respiration, but we also observe differences at the level of the mitochondrial P50. In fact, we observe for a global decrease of the mitochondrial of the COX P50, and especially at day 3. So that means that with a lower P50, the fish improve their oxyconformance at low oxygen levels. So they were more efficient to work under low availability of oxygen. So the main results in the liver is that we have a transient increase of the mitochondrial performance during the first day, and then the homeostasis came back. In the heart, it's another story. For the leak state, we also observe an increased trend for the leak state respiration. So again, perhaps a protection against the ROS overproduction. However, for the OXPHOS and also the COX, we observe a decrease of the respiration. So perhaps here in the heart, after 10 days of cyclic hypoxia, we have a decrease of the performance of heart performance. And finally, for the brain, we found, in fact, no differences. So perhaps here, in fact, we have a brain preservation because the fish could have a preferential irrigation of the brain, so to prevent the decrease of oxygen supply to the brain. And here, the brain would be preserved from hypoxia. So to make a summary of the results, what we observe here, for the liver, we observe a transient adjustment of mitochondrial physiology, perhaps to support the acclimation of fish to cyclic hypoxia, and then the homeostasis came back. For the heart, we perhaps have an eventual performance reduction, but more studies are necessary to see if we have effectively this reduction in performance. And for the brain, we have the preservation of this tissue. And finally, during the last part of this study, we want to know if you have any ROS overproduction in these three tissues. So for this, we measure the ROS level as a level of mitochondria in vivo. So we use the mitochondrial probe MitoB. So MitoB, in fact, react with ROS and produce MitoP. So if you have a higher level of ROS, you have a higher level of MitoP. And by doing the ratio between MitoB and MitoP, you can have a proxy for the ROS production. And in the three tissues, we found no differences. So at day 1 and day 10. So that means that we have no mitochondrial oxidative burst. Why? Perhaps it's due to the higher leak state, or we can have also the action of the antioxidant activities, which could buffer if you have a slight increase of ROS production during the reoxygenation phase. Other teams found similar results. For example, they also exposed some sculpin to cyclic hypoxia, and here again, they didn't show any oxidative burst. And I think also recently it was the same thing with anoxia and reoxygenation is the total. Using MitoB, it didn't find any increase of ROS production. So to conclude about this study, what we found, Arctic char, in fact, survive and acclimate to cyclic hypoxia by made some adjustment at the level of their metabolic rate. They suppress the spontaneous swimming activities to survive. However, they still defend the standard metabolic rate. At the level of mitochondria, we observe a lot of regulation at the level of the liver and a slight decrease in the level of heart and no change for the brain. And finally, we found no mitochondrial ROS upsurge in the three tissues. So the next step of this project for another day, or if you have a question about it, we characterize the proteomes of the liver to really understand which are the mechanisms behind this acclimation to cyclic hypoxia. So I would like to thank you for your attention, my team, and the people that give me money. Thank you.
Thank you very much.
Thank you very much, Loïck.
Oh, Elisa, you can you can do the questions round.
If you want, you can. No problem at all. Okay, so yeah, thank you very much, Loïck. It was a very nice presentation with very nice results. So do we have questions? Don't hesitate to just raise the hand directly on Zoom. But actually, I have one for you, Loïck. Oh, but Jason has one. So, Jay, you can go.
Really nice talk. I find the MitoB results really curious. And so I guess my question is it typical to see the same sorts of levels of production across tissues? Have people compared the MitoPB results, say, across tissues in mice or anything like that? Because I'm just not familiar with it.
I know that other people use this probe in other fish species. So in fact, it was developed also in Drosophila and mice, but I don't remember if the level were comparable in mice and in fish.
Yeah, actually, the follow-up question maybe on the ROS was were you able to test actually in real time ROS productions from your mitochondria in the Ouroboros assays as well, or is it something you purposely
No.
skip?
No, we didn't measure it inside the Ouroboros. In fact, we want to use this Metabolic probe because the advantage of this method is that you measure in vivo the ROS production. So because in the Ouroboros, you are in hypoxia for the mitochondria, and so this could perhaps have an impact on the ROS production. So it's one of the advantage, but yes, it would be also nice. We don't have the equipment during this project, but now Nico have it, and it will be perhaps nice to measure also if you have the same trend inside an Ouroboros.
In your run into anoxia would have been actually quite nice to see the correlation between your ROS production and PO2 in that regard. That was good.
Actually, I have a question as well on, oh, sorry, Jules. It's just more about the conditions you use with the normoxic and hypoxic phase. You said that the hypoxia was during night. So I was wondering if it's something that usually happen in the environment of the Arctic char, or if it was related to something else, because I don't know very well this species in particular. So.
Okay. So about this normoxic and hypoxic phase, so that's miming what happens in the environment, because during the day you still have the photosynthesis, so you have an oxygen supply in the water. However, during the night, so you have a decrease of you don't have the photosynthesis, but still have the respiration. So that's why we have this hypoxic period during the night. About this species. In fact, yes, for the most Nordic, so this species have a Nordic distribution. So circumpolar, here in Canada, the southernmost population are found perhaps in New Brunswick. We have some population in the Maine, but mostly in the north of Quebec. In Europe, you still have some population in UK, and in Russia, so you have some population. But so for the Nordic population, for the most Nordic population, I don't think there is cyclic hypoxia. However, for the southernmost population, where we observe an increase of eutrophication, you can have this cyclic hypoxia pattern in the environment, and with the increase of with the increase of the impact on the environment, and with the climate change, also with the rise of temperature, this species will have to cope more with this stressor.
Okay, I see. And I just also had another question about the sporadic increase in oxygen consumption that you observed only during the normoxic phase for the cyclic hypoxia fish. So you said that it could be related to like a suppression of activity to decrease the oxygen needs, like energy saving during the hypoxia. I was wondering if it could be related also to the fact that this increase in MO2, as you said, that could be related to spontaneous like activities, if it could be a sign of stress, like fish try to just escape this location, try to go somewhere else where they could be in normoxia, if you, as you say, that those fish are sensitive. So is it something that you could test maybe?
You mean, yes, you can test this. It would be nice, in fact, if you have a video confirmation for this study, but we don't have it. But yes, in fact, you can. I don't think the fish try to escape during hypoxic phase because we don't have this increase of MO2. We still have a basal MO2 oxygen uptake, so we just have the standard metabolic rate. And during the normoxic period, because sometimes we have to change the water and to make sure that everything is okay because it's only 10 days, the fish didn't see to appear. We don't see any special stress of the fish.
Okay, I see. Thank you. A lot of people wrote in the chat. Maybe if you want to say directly what you wrote.
It was more so regarding the mito-B, mito-P actually experiments.
Yeah.
Yeah.
Yeah, I just posted a link saying that they in Lao they measured mito-B by Rust production or Rust steady state actually in the mitochondria using this technique, and they get consistently the same result among tissues. And I see Jay's face, and he thinks it's suspect, and I don't know what to think about this.
I don't know if I'm suspect. I find it very interesting. The probe itself, I think the logic behind it's great. Consistency across does raise some questions on what exactly things like our H2O2 efflux measurements are actually telling us, if the in vivo levels are stable across tissues, but this is a different topic.
I gotta go, guys. Thank you. See you soon.
Cheers.
Thank you.
Actually, technically, it would be interesting to know whether the dye actually stays more in the endothelium space, because then if it just stays in the blood across tissues, then things would be a bit more consistent, I believe, rather than diffusing within cells. I don't know. Just some thoughts. A quick question, Simon, have you tried actually looking at beta oxidation or actually different substrate sources or even ketones or just different pathways or not really? Like, do you know whether the fish would actually burn more fat if they are stressed or if they are going under hypoxic stress or going under glycolysis, maybe a bit more?
In the mitochondrial, for the mitochondrial respiration?
Yeah, so for mitochondrial, let's say if you've tried like beta ox or even ketones. And
No, we don't try this. But I can show you what we found for the proteomic. In fact, that would perhaps be interesting to test. So for the proteomics, so after this is a change that we observed after 15 days of cyclic hypoxia. And we observe an increase of the protein dehydrogenase. So perhaps the fish use the protein as a mitochondrial fuel for the mitochondria after acclimation. So it will be perhaps nice to test it in the Ouroboros, but I don't have this result before. So perhaps for a next step of this project. If I don't know if I answer your question.
Yep. Yeah.
Okay. Yeah,
Jay,
Jason?
Yeah.
So sorry to jump in again, but I'm just curious, the metabolomics results that you alluded to, is there any clues in there? Like, did you find increases in succinate or anything that would show redox couples were showing any shifts with these treatments?
About the metabolic results, in fact, no, we don't see any increase of the succinate levels. However, as for the study, in fact, we sample our fish after the reoxygenation. So we don't see the succinate accumulation, if we have an succinate accumulation, happen during the hypoxic phase and during the sampling. When you sample after reoxygenation, we were not able to see any succinate accumulation. However, what we found is it's a decrease of the succinate after 1 month of cyclic hypoxia. We observe a significant decrease of succinate levels, and this could be a strategy, in fact, to counteract the succinate accumulation during the hypoxic phase. But we don't have the metabolism for the hypoxia during hypoxia.
I was going to ask exactly that question. Jay, yeah, I was going to say, no, it's good. I was going to interrupt you and say, following up on this. So you can't, even with the proteomics, you can't link back to what you found in the Ouroboros, but like for the liver, I guess proteomics is also on the liver. Yeah.
For the proteomic, what we only found for the mitochondria. So in this project, for the mitochondrial respiration, after 10 days, we do not have any increase. We have the homeostasis came back, and we don't see any increase or decrease of mitochondrial respiration. And when we look at with the proteomic results, we didn't see any significant upregulation of our mitochondrial complex. However, we saw an upregulation of the complex 3, and we also observed a downregulation of the ubiquinon biosynthesis process. So that means that the fish, after 15 days of cyclic hypoxia, they reduce the pool of ubiquinon. However, they increase the complex 3. And this could be a strategy to counteract if we have a reversal of electron transport. If you decrease the pool of ubiquinon and you increase the complex 3, the fish may decrease the reversal to the complex 1 of the electrons. I don't know if I am clear.
Yeah. Jim has a question.
Yeah, Jim.
Thanks. The questions that just came up were along my line of thinking as well. The decrease in succinate as cyclic hypoxia goes on is interesting, but you don't really see any proteomic. You don't see succinyl-CoA synthase or anything going down, or complex 2 going down.
No.
Which substrates did you use in the mitochondrial respiration assays?
Oh, yeah. So I will use, sorry for this. You succinate, to begin with, use some malate and glutamate, and not succinate. Oh, I don't remember.
Okay. It'd be interesting to know if flux through complex 2 changed, because it's fairly simple to change that. Post-translational modifications can change activities of that, and that can alter succinate metabolism, and again, maybe, like you were saying, prevent reverse electron transport if that's possible. So if you had managed to measure flux just through complex 2 using succinate, and whether it's leak or
yeah, oh, sorry.
No, go ahead. Then you could have a handle on whether that's going down.
Okay, I understand now. Yes, we add some succinate to measure the contribution of the complex 2. However, we didn't measure the complex 2 respiration alone without the complex 1. And yes, also about for the proteomic result, we didn't see any significant change at the level of the complex 2.
Right, but the contribution of even the contribution of complex 2 didn't change overall. So that's okay. So that's probably not a mechanism worth exploring further. Thanks.
Thank you, Tony.
Hi. Very interesting. I was going to follow on from Jim with that, and I was wondering if, first of all, whether you could have changes in the arrangement of your ETS. You know, if you could, if you thought about running, say, some native blue page gels to see if you're saying that you had an increase in activity with complex 3, maybe the arrangement. So if you're not seeing a change in the proteome, it's sort of similar to what Jim was saying. If it's a post-translational thing, it may not be a phosphorylation or something. It may just be an arrangement. So that could be worth a crack. That could be something to look at.
I'm sorry, I think I don't understand your last sentence.
Oh, sorry. I was just saying it's something you could look at. That would be a step forward.
Yes,
yeah. I'm not asking you a question. Sorry.
It's some Kiwi slang.
Yeah.
But these were in permeabilized fibers too, right? Or permeabilized tissue. So if you have isolated mitochondria, it's easy to run the blue natives, but if it's impermeabilized, then that's more tricky.
Yeah.
Oh, I'm going to jump in again. Loïck, did you do permeabilized liver as well?
For the liver, it was just a mechanical permeabilization.
Okay.
But yeah, it was a we permeabilized the sweet tissue.
Okay, because stuff that people don't often cite. We have a paper where permeabilization produces completely different results from isolated mitochondria, and it's if you know that the permeabilization does the same thing, that's great. If you don't have something to compare it to, or if the literature doesn't have something to compare it to, not that you have to do the experiment yourself, then you may be measuring artifacts. So it's
Okay, yes, it would be nice to have both as isolated mitochondria to compare with what we obtain from permeabilized tissues.
Yeah, and just in general, I mean, Eric loves to sell almost literally sell the permeabilized tissue model, but it's not appropriate in all cases. It can be a great tool, but you just have to make sure it's the right tool for the question you're asking.
Okay.
Great.
Thank you. Do we have other questions, maybe? Actually, I just
look
have
at
one about.
yeah?
Oh, sorry.
I have a quick one. Sorry, Elisa. Did you look at the
No, you can
proteomics
go.
in the heart as well, or just in the liver?
Can you repeat? I misunderstood.
The proteomics work that you started
No,
mentioning.
just
Okay.
no, just in the liver. We
No, I was
measure.
wondering.
We will measure the DNA methylation in the heart, but we don't have the result yet.
So why do you think OXPHOS decreases in the heart with cyclic hypoxia? Do you think it's because they're not getting that training at night, those bursts? Did you see the fish behavior also change? Do they swim less, like in the normoxic, like in the day conditions? Does that affect their behavior?
Yes, I think we have an impact. I just have observational, I just have some observation, not really measurements. So they seem to calm down during the hypoxic phase, and with what we observe for the metabolic rate measurement, clearly with the suppression of this sporadic increase of MO2, the fish decrease their activity, they decrease their activity, they calm down. About the art results, in fact, it would be nice to measure with this paper, we can't really conclude if it has an impact on performance. So it would be nice to measure the heart rate and other heart measurements to really know if we have if the sensitivity of Arctic char to hypoxia is mainly due to the decrease of heart performance.
And content, mitochondrial content as well, I guess.
Yeah,
no?
yeah.
Cool.
Yes, Jason?
One last real quick question, then I got to run, but looking at those declining heart respiration values, did the heart swell, or did you happen to measure the size of the ventricles with the cyclic hypoxia?
Can you repeat the last sentence, please?
Sorry. One response that can be to compensate for declining metabolic capacity is increasing heart size.
Okay. No, we didn't. I don't measure it for this paper, but for the first one. So when we expose fish up to 1 month of cyclic hypoxia, we didn't see any relative change in mass of the heart. So no, the heart didn't become bigger.
Yep, that was my question.
Yeah, about the heart performance, actually, it would be super nice maybe if another project arrive after that, just to see if there is something at the tissue level or at the individual level that could show that there is an impact on performance of heart, because usually you would expect the reverse, like an increase in the heart performance to increase the oxygen transport to better acclimatize. But that's true that this species is considered as sensitive. So potentially this decrease in heart metabolism could show why they are sensitive, because they are potentially not able to maintain their heart performance.
Yes, that would be very cool to investigate this more. It would be so nice to see if we have any impact on the maximal metabolic rate, but yes, it would be for another student.
Just a hint to junior researchers out there. Measuring tissue mass is one of the easiest things to do. So whatever you're doing, before you go on and isolate mitochondria or permeabilized strips, measure the friggin' mass, write it down in your notebook. You may never use it, but it's super easy.
Yeah.
True.
On that, so you did some proteomics. No, you did methylation or something on the heart, wasn't it? But no proteomics.
No, we
Is that
don't do the proteomics on the heart, no.
Yeah. So that would be somewhere to look going forward as well, because I've done work on mammalian diabetes and cardiovascular issues with high blood pressure and things like that. And you get isoform switching with complex 4, which changes its affinity and activity. And with hypoxia, it tends to go down as well, the activity in mammalian systems. So that could be worth a think there.
Yeah, it would be nice. Perhaps we will have some answer with the DNA methylation, but yeah, we don't have. We have to choose one tissue for the proteomic project, and we chose the liver because a lot of things happens at the level of the liver, and the aim of this the last part of this project was to understand the mechanism behind the acclimation of fish and how serious they do it. But yes, it would be nice, perhaps in the future, to do the same for the heart. Yeah.
Mm. Perfect. I think that Loïck had plenty of very interesting questions on this article. Thank you so much, Loïck, for the presentation.
Yeah.
Well done for this publication. You can be very proud of it. So yeah, if everyone is okay with that, I think that we can say we're done for today. So thanks again, Loïck. Thank everyone to be there for this very nice presentation. And as usual, we will send you the link of this meeting very soon, and an email for the next Mito Club that will happen in November very soon as well.
A proper link
Yeah.
this time.
Yes.
Not a double link, just a single link.
Oh yeah, no.
And also, I don't know, I opened to everyone with a Zoom account, so it shouldn't have been an issue to log in. I don't know what I did, but I can pay more attention next time.
Yeah,
No
I
problem.
just
worked
wanted
in the
to
end.
say that the German site could not log in there easily. I had to sign in into my account first, and only then I was allowed in, and Amanda was not able to come in at all. So
I got her email.
if there is that issue again, you can let me know and I can make a link. It seems that the University of Rostock lets everybody in without asking too many questions.
Okay. Yeah. Let's. If you want to try to connect maybe 15-20 minutes earlier and see if it works. And if it doesn't work, send me an email.
Okay.
Because I think I got the right. I think I selected the right option. I will check again.
Okay.
Because
anyways,
with Jules' links worked with everyone, I think, no?
Yeah, I can.
Is it only mine?
The Auckland Uni one, we can try with Auckland Uni next time.
Yeah,
as
we
well.
can try with a different. Maybe UCL is very very
restrictive.
private.
Restricted.
Well, it could be
anymore.
Yeah, it could be the licensing or something of Zoom, because I think Auckland Uni has quite a broad license.
Yeah.
Hey, thanks very much, guys, for organising this.
Okay.
Once again.
Thank you.
thank
Thank
you
you very
bye.
much.
Thank you.
See you soon. Bye.
See you.
You guys.
I'm gonna
Merci,
stop
Loïck.
the